Okay, we are still in section 1, the mechanics of DNA, and this is the Genomics Revolution section of the part of this section. I've already mentioned a genome several times, and a genome is all the DNA in an organism, including its genes. So it's just not the sequence of its genes. It's all of the DNA, and I've already mentioned that there's a lot of DNA in humans that is not coding for proteins. Genes carry the information for making proteins required and therefore control development, as I've just explained, how we go from genome or from gene to genome. Now, in reverse, what we're interested in doing is going from genome to gene. How do we sequence that DNA, and what are some of the tools that we need to understand how that process is done, and what will this technology come to bear? Remember that all of the DNA is replicated in every cell every time a cell divides. So your 3 billion base pairs of DNA is copied every single time a cell undergoes division. In genomic technologies, we will break this into really 3 levels. Genomic sequencing, sequencing the DNA. We can look at measuring RNA levels. This will tell us in any given cell what genes are being turned on. You can imagine that the RNA levels will be different in a cancer cell from a normal cell. What RNA populations are being transcribed will tell us a lot about the metabolic function of that cell. Also, similarly, measuring the population of proteins. Not all mRNAs are going to be decoded into proteins, and not all proteins are going to be functional. So therefore, we have genomics, which is the study of DNA. RNA is called transcriptomics, and the study of protein ecology is called proteomics. So we've already described that I can take his arm and put it in a Waring blender and isolate his DNA. We've done that several times during the past few lectures already, and it would look something like this. In fact, you are all going to isolate your own DNA towards the end of this course out of cheek cells. And it will look something like this. It's this white flocculent material. We know that the structure of DNA is this sequence of A's, T's, G's, and C's, which contain this information. So the structure of DNA tells us a lot about its ability to contain information. And in a human, or in lots of eukaryotes, the DNA in the nucleus is packaged into chromosomes, and in humans we have 46 chromosomes, 23 from your mother and 23 from your father. And two of these are sex-determining chromosomes, an X and an X if you're a female, and an X and a Y if you're a male. And remember, as shown here from your chapter 5, that deoxyribonucleic acid DNA consists of these base pairs designated A's, T's, G's, and C's. A's always pair with T's, G's always pair with C's, no matter what side of the helix that they're on. So how would we go about sequencing 3 billion base pairs? First of all, I've got your DNA in a jar now. Well, that's great. Well, how are you going to, what am I going to do? Take a microscope and look at his A's, T's, G's, and C's? I don't think so. So elegant sequencing reactions were first developed really by this guy named Fred Sanger. He devised elegant chemical methods for sequencing proteins, but he also developed methods for sequencing DNA. And one of the methods that he first developed is shown on this slide, where if you have a gene that you want to sequence, let's take his hemoglobin gene again, and we have a primer that will recognize some sequence on that single base pair strand, shown here in the bottom left. And if we make that radioactive, I can make that primer radioactive by using P32. It's a radioactive phosphate. So if DNA contains a phosphate backbone, I can make that backbone radioactive and then recognize part of his hemoglobin gene and put it there. So now we've got that's hot. I can detect it with a Geiger counter. Now there are these strange analog bases. Rather than just being A's, T's, G's, and C's as deoxyribonucleic acids, these are analogs called dideoxybases. And if they go into a developing strand, it'll stop replication. So Fred Sanger used these dideoxys for sequencing. If you randomly include these with a polymerase that would extend that radioactive primer, as shown here, if you stick a dideoxy A next to that T where it's going to go in there, the reaction stops. If a normal A goes in and the next one that goes in is a G that's a dideoxy, it'll stop there. Therefore, if I run this reaction all at the same time, I'm going to generate all these different lengths. Now I run them out on a gel, I put a radioactive detection film over it and expose it, as shown in the results on the right-hand side of this slide. So you read this gel backwards. So look at A should go in here, G should go in here, C should go in here. So here the dideoxy A, you start at the bottom, which is the smallest fragment in the gel, right? Remember how gel electrophoresis works? Small fragments go to the bottom, large retained at the top. So that's A. The next one is G, as I predicted, C, and you can read what it should be, and you go upwards here to detect this. And this is how the first sequencing reactions were done. Manually sequencing the first genome of Haemophilus influenza bacteria, done this way. They would pour these acrylamide gels in between window pane glass that was roughly about this big and run this thing, and here's an example of one of those gels shown off to the right here, and it would be this radioactive sequence that somebody would be looking at. It'd peg a Geiger counter, you know? And then they develop these films as shown here and read them backwards. So now the way that this has been extrapolated, now you know the mechanics of it, rather than dideoxy bases, each one of them is a fluorescently tagged base. And robotics have been used to detect these going through at very rapid rates. And now you get a readout like this that's shown here on the right-hand side. And you can do these high throughput genome sequences that would be really read in capillaries that might look something like this. And it was the development of that technology that led to the first draft of the human genome, the draft, published in February 15th, 2001. The final version was published in 2004. And as I mentioned previously, this accomplishment far eclipses that of landing on the moon. In fact, you know, you could say that landing on the moon was a great accomplishment, there's no doubt, but it's a rock, you know, and it's not going to change your life that much. It has, I mean, it brought us technology, but knowing all the genes in humans will have a big effect on a lot of things going forward. The mouse genome was subsequently sequenced in 2002. More recently, in the November issue of 2011, the mac monkey. The rice genome was sequenced also in 2002. So now we know the genome sizes and the genome sequences of many organisms. I should point out that genome size does not well correlate with organismal complexity. For example, if you use genome size as sort of a genetic IQ, humans are not doing so well in comparison to an amoeba. Same thing goes with the number of genes. The number of genes in a human is probably about 2% or maybe a little bit more than of its entire genome. A fruit fly is 19%, but the E. coli is roughly 90%. So how do we use genomics to understand relationships between organisms? Well, we know that the rice genome was published in 2002. I want to start off this with a recent example that one of my graduate students just finished up. He was doing his master's degree, so this is what it takes to do a master's degree in this field. He looked at the characterization of genes in rice, which have similar sequences in corn and maize, which determine sexual development of the flowers. Maize is a monoecious plant, meaning one home that has the male part, which produces the pollen at the top, and the female part, which is the ear, as an axillary on the side, as a lateral. There are some plants that have two homes, male and female on different plants, like a ginkgo tree or various conifers or hemp. Sex determination in maize. How does the male become the male and the female become the female? Roughly, they all start off as perfect flowers. The male and the female primordia are in the same flower in maize, and when an ear develops, the male is aborted. When a female develops, the male is aborted. When the male develops, the female is aborted. There are mutations in both of those genes which regulate that pathway, which a friend of mine at Yale is an expert in and that my student studied with. He used bioinformatics to compare these and looked at promoters and termination signals in rice and maize. I'm showing you an example here of how modern genomics can be used to study a developmental pathway like sex determination in plants. So he did this comparison between these genes which are involved with male and female development and show clear comparison between maize and rice as they have diverged from each other only a few million years ago. So in other words, the genes that cause male development in rice are also present in maize and the other way around. And that sometimes this homology is widespread across those genes, upwards of 82% in this case. What I mean by this is by this example, we can take any organism and do this. This is maize and rice. We can do this with humans and chimps. We can do this with humans and fruit flies, humans and mice, humans and rice, and look at how sequence comparison relates to genome complexity and how genes are regulated and turned on and off. In one of these experiments, we've crossed rice with another grass and made a hybrid trying to make rice monoecious the way maize is. You could say, like, why would you want to do this? I took this picture, and I'm rather proud of it, just a couple of weeks ago, in fact. This is a picture of the flower between that cross. I was at the imaging center at Brown. If you take a bunch of these all in one day, you start to have them in your dreams, which isn't so bad. But genomics. Now, high-throughput genomics allows us high-throughput sequencing. Make no mistake about it, the Beijing Genome Institute is going to sequence everything. Realize what that means. What a powerful resource, first of all, but being able to use that in genetic outcomes, using that on human populations. And as you know, knowledge is power. So understanding the evolutionary relationships among species is made possible by these sequence comparisons. I just showed you what my master's student is capable of doing. Realize where we are now. I don't mean to put him down, not by any means. He did a great job. But look at what we're capable of doing is what the point is here, right? So if you look at the mouse genome mapped on the human genome, we can take all the mouse sequence and compare it directly to humans. You're 88% the same as a mouse. And here's exactly where those genes overlap. If I wanted to, I could show you right down to the resolution of A's, T's, G's, and C's where you're different and where you're the same as a mouse. And we can do the same thing with the chimpanzee, and there it is. So if you ever wonder how close you are related to some of these primates, you can just look that up. You can do what's called a blast search. Put that into Google, B-L-A-S-T, and compare any genome you want. All capitals. 95% of the chimpanzee genome maps identical sequences to those in the human. And using the sequence analysis, this kind of ancestry can be well documented. So how can we use genomics to see evolution in the human genome? As I mentioned, humans are 46 chromosomes. And here are some of the bases that make up those different chromosomes. Like chromosome 14 is 109 million bases, chromosome 18 has 85 million bases, and it's that variation that is considered significant. Since I'm harping on your hemoglobin gene, why don't we not stop? Hemoglobin, the globin genes are present on humans at a locus in chromosome 11, shown there, and that the beta globin gene locus on chromosome 11 actually does comprise 6 genes. One of them, as shown here to the far left, is on during embryo development, only during embryo development, regulated by a promoter. This then shifts to those two shown adjacent to that that are on in a fetus. This is interrupted by what was called a pseudogene. This is a non-functioning gene. It has the sequence of a globin gene. It just doesn't make globin, okay? I'll get back to this in a minute. And then there are these two genes that are shown to the right here that are on in an adult. So actually you do have 5 copies of this gene, actually 6. One of them just isn't functioning, and we call that a pseudogene. So you have 5 active genes expressed in the sequence in human development and this one pseudogene. If we would do isolation and nucleotide sequence analysis of the beta globin pseudogene from human, gorilla, and chimpanzee, you can match those all up. Striking what this shows. That why doesn't the pseudogene work in humans? It also is exactly the same sequence as the one in chimpanzees. Exactly the same. Another interesting part about it is these mutations. There's a whole bunch of them. It's not just one. We have one that's altering the control region in the promoter. We have a defective start signal. We have stop signals that are in that first intron. 3 stop signals in the second exon, a frame shift deletion in the first intron, and a frame shift deletion in the last exon. Now, check this out. If we do a sequence comparison of the same pseudogene for beta globin in the chimpanzee, panpeniscus, we see that the chimpanzee has the same identical matching mutations. So now we have the chimpanzee genome and can do those kinds of analyses. Where did humans get their 46 chromosomes? Our most closely related primates have 48. So we could look at evolutionarily how this might have happened with a testable hypothesis that if these organisms actually do share a common ancestry, the human genome must contain a fused chromosome. So in fact, the human chromosome complement has 46, and chromosomes have characteristically these centromeres and telomeres. Panpeniscus, the chimpanzee, has 48. So we would imagine that a fusion could be detectable by the fusion of 2 telomeres as shown here. Chromosome 1 and chromosome 2 might share these 2 telomere fused sequences as shown there. In fact, human chromosome number 2 shows the exact point at which that fusion took place. So the sequence and the junction of those shows the fusion of chromosomes 1 and 2 in Pan-Paniscus that resulted in chromosome number 2, or chromosome 1 and 2 in humans that resulted in that second chromosome. So we can use comparative genomics to look at relationships between not only humans and other primates, but humans and all other organisms. How closely related are we? A gene that is homologous in everything from pea plants to humans has a very similar sequence. Also, we now have the entire Neanderthal genome. So not Homo sapiens, and also the Denisovans, another branch of humans that was genome sequenced just last year. And we can line these up and look at the lineage of humans and the comparison of admixtures that came from those different organisms. So first of all, sequencing technology has itself come a long way since the days of Fred Sanger and manual sequencing. There are companies like Illumina, HiSeq, and 454 that have really pushed the envelope in high throughput genome sequencing to bring the price down from the $1 billion that it costs to do the first draft of the human genome down to what we're seeking now as under $1,000 and probably could be completed in less than 10 days. So especially companies like Illumina, HiSeq, and 454 have brought this technology to the forefront. And some of what this involves is shown here on this slide, where breaking up the sequence into very small fragments and attaching it to beads that are then placed in these micro wells, and the sequence is deduced off of these small beads and then put back together using algorithms. Now, I want to make a point here that this is kind of like if you were to take an old-fashioned telephone book from Manhattan and cut out all the names and just throw them up in the air and put them all back together again. That's what this algorithm is all about. And I don't know if you've seen that old movie, Jurassic Park, and remember how that happens. Presumably, the conceit of that movie is that a mosquito landed on a dinosaur, right? Any of you ever seen this? Right? The mosquito lands on the dinosaur, sucks some dino blood, and then a gob of resin overtakes the mosquito. It's amplified. Some guy millions of years later goes in and takes the blood out of the dinosaur, which contains the genomic sequence of the dinosaur, right? And a lot of people, when that first came out, said, oh, these people in Hollywood, that's ridiculous, right? The DNA in that amber is just going to get all chopped up into pieces. How in the world are you ever going to put this thing back together? And that was the criticism of Jurassic Park when it first came out. Well, lo and behold, that's the way it's done. We don't just wait for nature to fragment the thing up, we do it ourselves. We fragment it all up into these pieces, and then using sophisticated algorithms, piece the entire thing back together. Do I mean to say that it might be possible someday to deduce the sequence of dinosaur DNA? We just did it for Neanderthals out of a fossilized femur bone. If you can get DNA, it doesn't have to be in pristine shape in order to deduce a sequence because of this kind of technology. And even going back to that, remember they stitched together the dino DNA with frog DNA, and that's what presumably caused the problems that we saw in the latter part of that science fiction movie. But actually, that is fully capable with the technologies of genetic modification that we carry out now rather routinely. So recovering ancient extinct species and their DNA, I wouldn't say is totally out of the question. Would it be possible, for example, to sequence mammoth DNA to put the whole thing back together and maybe implant that into an extant elephant as a surrogate? There are people that are pursuing that. there have been extinct species that have been recovered already. There was an extinct cattle called a bantane, which was recovered, and now there have been 2 bantangs born in the San Diego Zoo to surrogate cattle, much along those lines. So sequencing technology has gone a long way, and this is going to play a large role in the subsequent lectures that we look at the different technologies and applications in biotechnology, from agriculture to pharmacy and medicine and so on. One of these involves pharmacogenomics. So you see the word genomics, all of the DNA in humans in this case, and pharmaco, the first part of that, pharmacy. What this really refers to is looking at your DNA or an individual's DNA and determining how drug metabolism may play a role in health. For example, a drug that might be good for you could kill him. And wouldn't that be good to know ahead of time? Or a drug that might be good for you might not do anything for you. And very well, this may be represented in your genome. So by doing genomic analysis, we can do personalized medicine and determine not only disease diagnostics, but what drugs might be appropriate for certain individuals and not others. So personalized medicine has come out of a branch of pharmacogenomics in the idea that we can base an individual's DNA on treatments. So having affordable DNA sequencing and analysis of the 6 billion base pairs of human DNA will allow this type of diagnostics and treatment. So the beginnings of this you just start to see now with diagnostics for certain cancer types and so on. But we can also see that this has, while tremendous potential, it also has tremendous downsides. You could learn, for example, at your age, that you have a gene that not only predisposes you to a disease, but that one you will get. Say Huntington's disease. If you had 2 recessive alleles for Huntington's disease, you're going to get that disease. And there's no cure for that. Do you really want to know that? On the other hand, it could show you that you have a predisposition for type 2 diabetes. If you start treating yourself correctly now and live a proper lifestyle, you could avoid that. So on the one hand, personalized medicine and genomics might influence lifestyle changes. On the other hand, it could cause a lot of angst. And also, we could say there could be a lot of false positives. What if this gene shows that you have a 30% predisposition to breast cancer? What are you going to do with a 30% number? And what could you do to avoid it anyway, even if you could? So disease predisposition, disease diagnosis is a double-edged sword in this case. It will inform us a great deal, and in my opinion, knowledge is better than ignorance. And I think the more people that have access to their genome, the more sort of Wikipedia-like genome analysis will become. So the ultimate genetic test, as referred to here in last June's issue of the journal Science, shows that the encoded DNA sequence of our individual genome will build, maintain, and adapt all our tissues and functions, and this knowledge will help us make appropriate choices in many cases. This also informs the debate about who we are and where we came from. We're a diverse world in many ways. We have more than demonstrated at this point that all humans are derived from African origins, and that the admixtures from certain break-off groups that have recontributed to the genome has ultimately resulted in our current population. You could fast forward this with now populations no longer geographically separated, that we are becoming rapidly a global population. You can even look at what the eventual composition of humans in 100 years might look like based on the phenotypes of combinations of people who contribute to our lineage. This also informs us a lot about race as being a cultural phenomenon more strongly than a genetic phenomenon. Skin color, for example, is one gene. IQ, generally as a quantitative trait locus, turns out to be nearly the same globally. So you have a global situation based on IQ that is relative to the human genome, but these individual small characteristics that sometimes some people make way too much of. So a few techniques. One is a technique called Southern blot analysis, which was invented by a guy named J.D. Southern in 1975, hence the name Southern blot. And this allows for specific detection of DNA sequences. And even though this was invented in 1975, it is still standard fare in most laboratories, including our own, for detecting specific DNA sequences. So let me show you an example of, first of all, how this is done, and then how this might be used. I keep picking on this guy's DNA, but as I said, I could put him in a wearing blender and isolate his DNA in a matter of an hour or so, and I would have DNA from this individual that then I could then analyze. And we've been picking on his hemoglobin gene, so we might as well keep going with that as an example. But we will all isolate our own DNA in the last class, so I can show you how to do that. And you can walk out with a sample of your own DNA and give it to a close friend or a relative if you want. But it's a rather simple procedure, and we can isolate DNA from any living or extinct organism, and I'll show you how to do that in the last class. The Southern blot uses a medium called nitrocellulose, which is a membrane which we can bind DNA, RNA, and proteins to affix them to this membrane and then use probes to analyze it. The membrane has these properties where it's hydrophobic, that means it repels water. It doesn't bind double-stranded DNA very well, so we separate the strands first, and it doesn't bind small DNAs very well, or large DNAs. So typically when we work with DNA samples in Southern blots, we like to chop them into chunks. And these chunks we would preferably like to have around 20 kilobases or so. So the way that we would take his DNA to analyze for his hemoglobin is we would first either randomly shear it. Like if I take his DNA and it's a big gob that looks like this, and I vortex it or shake it really hard, I can randomly shear that DNA into smaller chunks. Or I can use an enzyme to cut it randomly, the way I showed you before for cloning. We can use restriction enzymes that will cut randomly in the genome, and then I can take these cut samples and using gel electrophoresis, run them out the way I showed you before, where the DNA will migrate towards the positive pole because it's negatively charged, and the larger fragments will stay at the top. The smaller fragments will run towards the bottom, and because there's so many of them, it'll create this smear. If I take that gel, the word blot comes from what I'm going to show you how to do next. You take that gel out of the gel electrophoresis unit and set it onto this platform, which I show in this diagram here. And this is in this container in which we can put a buffer. So there's the gel. It's labeled right there. That's on top of a blotting paper. Typically, it's a Whatman filter paper that allows the liquid from the buffer to wick up underneath the gel. So on top of that, I place the membrane filter, the nitrocellulose, and then a stack of paper towels. This might not look very sophisticated, But this is exactly what we do in the lab these days, as it was first described by Southern in 1975. And then we use a sophisticated weight, perhaps a textbook or a brick or something like that. And what happens here is that the liquid that is drawn up by this blot paper then is transmitted through the gel, and the DNA is adsorbed onto that nitrocellulose filter where it sticks. And we've already soaked that gel in something that would make it single-stranded, so that the single-stranded DNA now is stuck to the blot. So this is a way of transferring all of the DNA from the gel onto the blot. That'll take overnight. We leave that overnight, go home, watch a movie or something, and we come back. And now if I want to find his gene for his hemoglobin, presumably it is somewhere in that smear that is now on the nitrocellulose. I take that nitrocellulose out, I put it into a Ziploc bag, or a sealamine bag, if you know what that is, and we will put in a hybridization buffer that contains a radioactive probe. Let's say I have a probe that is labeled with P32. It's hot, it's radioactive. Its sequence is complementary to his hemoglobin gene. The only place it is going to hybridize is going to be to his hemoglobin gene and nothing else. I then rinse off all of the radioactive probe, which I can follow with a Geiger counter, and then expose that blot to a film. The film will only expose where the radioactive sequence is, and that will detect his hemoglobin gene. See that? I can find any gene I want that way. And this is especially relevant to, let's say I genetically engineer this guy, and I put in some gene from some other organism. It's hard to do now because he's an adult. But basically, let's say I already did that a long time ago, and he's got a foreign piece of DNA in there. Or he's genetically engineered. Or let's say I have a genetically engineered plant that has a gene in it from a microbe. I can find that gene specifically by using Southern blot analysis. Everybody follow that? I can find specific DNA sequences by this method. So again, just to be clear, these are the steps involved. You separate the DNA out on an agarose gel. You denature it with a base, like sodium hydroxide, that makes the single-stranded DNA. We then neutralize that, so it's not basic any longer. And we equilibrate the gel with this buffer. This is a famous buffer that's used for that purpose, SSC. We then transfer it to the nitrocellulose, pre-hybridization buffer, hybridize with the radioactive probe, wash the filter, and expose it to an X-ray film. Since radioactivity isn't so user-friendly, we get a little paranoid about these things these days. Colorimetric, non-radioactive methods have been developed, and these are just as sensitive. And we also use luminescent probes now, so we can pretty much get rid of the radioactivity. We don't use radio-labeled probes hardly ever anymore. So a Southern blot then is used for, give a shot at it. Okay, that's good. Detection C is right. You guys, A and B. This is not for sequencing. I already showed you how to do that. It's not for detecting proteins. This is for detecting DNA sequences. Answer C. And look at that. You're pretty much the same as last year's class. So I mentioned that nitrocellulose can also bind RNA. So if we took all of the RNA, instead of isolating his DNA, if we isolated the total population of RNA, and I wanted to find out what genes are you expressing? Out of your 24,567 genes, What are you expressing in terms of making RNA off of? Are you making 24,567 of them? Or if I were just to take RNA from your eyeball, what set of genes is eyeball specific? We could do this with tumor cells too and say, oh, you have a tumor? What genes is that tumor actually expressing? So the same technique, but instead of running out the RNA onto a gel, instead of running out the DNA onto a gel as we would in a southern blot, we run out RNA. And because the southern blot was named for DNA, they very cleverly named this a northern blot. And that's how this got its name. But it's essentially the same technique. And this has now been reconfigured to do in these sort of dot blots as shown here, now called microarrays. And I will talk more about those in another lecture. Now I also mentioned that we can detect proteins by probing for proteins also affixed to nitrocellulose filters. So in this case, instead of isolating DNA or RNA out of this fellow, I take all of his proteins. And I can run all of these proteins out in a gel. And let's say I wanted to find one specific protein. Now I can't do that with a probe, can I? How am I going to find one particular protein against the ocean of proteins that this guy is making? Let's say I wanted to find a specific I protein from this guy, and I make a homogenized version of him and do a gel. How do I find one I specific protein against the hundreds of thousands of proteins that make up this fellow? Well, we use antibodies to do this. Now this is a brief introduction to antibodies, and this will come up later when we talk about vaccine development, and a little bit about the immune system right now might be helpful for this. Whenever you encounter a foreign substance, a foreign antibody, a foreign protein, a virus, a bacteria, by any mechanism, the first reaction is going to be by your white blood cells recognizing that something strange is in me, or in you in this case, and react to that. Your immune system then will generate antibodies, these proteins, that are specific to whatever you encountered. This is absolutely amazing. Considering the vast amount of stuff that you get exposed to during your lifetime, your body is capable of making these unique proteins against whatever it is you encountered. This gives a backdrop to how vaccines work, but basically, if I were to inject you with a foreign protein from, say, a corn plant, I can come back and 10 days later, isolate an antibody from you that was specific to that protein that I injected you with. This is incredible. I can do this with mice. I can inject mice with a human protein, say human insulin, and then 10 days later I can go back to that mouse and I can isolate an antibody that will stick to what? Human insulin. Okay, you got the picture? I can make an antibody against anything. And your cells are going to retain a memory of that. That's also amazing. So these antibodies look like this Y-shaped protein conglomerate that's actually not one protein but several that are stuck together. So again, in this poor individual that got this nail in his foot, some bacteria were introduced, and from this, these phagocytes recognize the foreign invader, and subsequently antibodies are produced that signal white blood cells to take care of that, but antibodies then are in your bloodstream. So antibodies are proteins coded for by genes. There are a set of genes in mammals, other organisms too, that code for antibodies. And in this diagram, you see these genes are comprised of V segments J segments and C segments. And there are variable chains that are capable of making different proteins specific to foreign proteins that are introduced. So it's this sort of shuffle the deck idea where you can make a specific antibody to virtually anything that you're going to encounter. So we can make mouse anti-human insulin, for example, or mouse anti-human hemoglobin. You're famous, I'll tell you. So these antibodies are these versatile proteins, and we can engineer these too, but we can use these to recognize any antigen, and an antigen is any foreign protein. Some of them could be pathogenic. And these are presented on the outside of B cells in your blood. One way that this is used, if I go back to my mouse example and the insulin, is if I inject human insulin into this mouse and about 10 days later take out its spleen, I can isolate these B lymphocytes from the spleen and fuse these to these cultured myelinoma cells. These cells then have, they can be cultured perpetually, so we can fuse the mouse spleen B lymphocytes that contain antibodies against human insulin to these myelinoma cells. Those cells are called hybridomas, and when we grow them in culture, they will produce antibodies against human insulin. And we can grow them in culture and make a lot of anti-human insulin with these monoclonal antibodies. The reason I'm showing you this right now is this is the backdrop to how many, many drugs are now being made, making monoclonal antibodies against human proteins that are involved in diseases. I'll show you an example of a drug that's made just down the road from here at Amgen for rheumatoid arthritis. In rheumatoid arthritis, humans make, it's involving the tumor necrosis factor alpha protein, and you're making antibodies against your own tumor necrosis factor alpha. It's an autoimmune disease. And so by interrupting that with monoclonal antibodies, you can disrupt that disease. And there are monoclonal antibodies that are made against human cancer cells to disrupt their division. So I think you will see that this particular technology is very useful in manufacture of those types of drugs, but it's also very useful in making antibodies for a Western blot. So how a Western blot works, so we have the Southern blot for DNA, the Northern blot for RNA, and the Western blot we run out proteins onto this gel and then blot them the same way. And now, in order to find the insulin, or in his case his hemoglobin, if I had an antibody to his hemoglobin and tag it with a fluorescent dye, that antibody is going to stick only to his hemoglobin. And then I can see that as a fluorescent tag and detect his hemoglobin. Has anybody ever thought of another use for this? How can we use antibodies to detect things that might be useful rapidly? Let me show you this. So a Western blot then is used for detection of proteins. One way to do this is to take your monoclonal antibody. Let's say that monoclonal antibody was against human chorionic gonadotropin, HCG. And we attach that monoclonal antibody to a plastic substrate. Let's say that's a dipstick. Let's say it's the bottom of a well. And then the specific antigen that finds that might be the human chorionic gonadotropin. HCG is produced very soon after pregnancy in humans. So you can use this as an early pregnancy test where the protein is present in blood and in urine, and will find that monoclonal antibody and is very, very specific and is very, very reactive. So if we then use a secondary antibody that sees this, then we can increase that specificity and attach an enzyme that allows that to be colorimetrically deduced very quickly. So you can develop an early pregnancy test with a dipstick like this in really only a matter of minutes and show whether or not a person is pregnant really only days after fertilization. We can also now, this has been developed for home HIV tests, which was a long time in coming, I think, but lots of other applications where, say, you might need to detect a protein very rapidly. And very specifically, you can detect very small numbers of molecules with this type of an application. And so you can think of lots of ways in which this enzyme-linked immunoabsorbent assay, or ELISA, technique might be useful. I want to get a little science fiction on you at this point before we end this section on Genomics Revolution. And this isn't science fiction. In fact, it's something that's just, it almost seems science fiction, but it is something that's happening right now. So I have told you already that we can cut and paste DNA to clone DNA. We can take a promoter and fuse it to a coding sequence and a termination signal and make a transgene that we can then move into other organisms. No problem. We do this all the time. We can cut and paste DNA since 1974 or so, 75 or so, since Boyer, Cohen, and Berg did it. What I'm going to show you is current day DNA editing. This has now really been bumped up a notch. So there are two techniques that have just emerged in the last few years that allow exquisite DNA editing functions. So these are new cut and paste functions that are not nearly as laborious, but also involve specific sequence targeting. One of them is called zinc finger nucleases. I know that sounds bizarre, but I'll show you in a minute what these are. These are DNA binding proteins that are capable of recognizing sequences of 3 bases. And then there are these other ones called transcription activation-like effector nucleases, or we call them TALENs as an abbreviation. I mean, I know that's weird, and you might think this is out of the context of this course, and this might be normally presented in a more advanced course on biotechnology, but I'm doing it here to show you what the state of the art is and where this is likely to go. So I do think it's relevant in this class. So first, zinc finger nucleases. These are now being sold. This ad comes from the back of the journal Science called BioAccess. It's sold by Sigma, in which you can buy zinc finger nucleases which will target any sequence you send them. And you send them a sequence of 9 base pairs, they'll send you a zinc finger nuclease that'll target that. That's their business. It's a reagent. And how does this work? A zinc finger nuclease is a protein that's capable of recognizing 3 base pairs. And if you say ATG, you can design or isolate one of these things that will recognize and bind ATG. Now the problem is with that is that that sequence is redundant in your 6 billion base pairs all over the place. So that's not much of a use, If you had another one that recognized CAC and you had a 6-mer, that's pretty good. Your resolution's getting better, but ATG, CAC is also kind of redundant. You're going to see that thing all over the place. So let's make a 9-mer. Now you're getting pretty unique. If you make a unique 9-mer, as shown in this top diagram here, you're getting fairly unique. Now let's link that to an enzyme that's capable of cutting DNA right at that site. So there is this nuclease, this cutter diagrammed here, that actually functions as a dimer. It doesn't function when it's just one piece of it. It needs both of them brought together. So if you have a 9-mer over here and a 9-mer on the other side of your target that brings in the dimer of this enzyme to cut, you can determine exactly where you want to cut anything. If you know that sequence, his hemoglobin gene, if I know the sequence, I can devise a zinc finger nuclease to cut it wherever I want to cut it. We have the sequence. I can cut this thing wherever I want. Let's say I knew a DNA sequence in humans that caused cancer. I could cut that right there. What function would this have? Well, when you cut DNA like this, oh, by the way, that enzyme is called FOC1, and this is pretty much what people use all over the place. So this is the zinc finger nuclease here attached to the monomer of FOC1, and the other DNA zinc finger nuclease is here, recognizing that sequence, brings in this other monomer, makes the functional dimer, and cuts it. So essentially, there's the situation there, and there's the sequence that's cut in the middle. So what ends up happening there are two possibilities. You cut that DNA sequence right there, and if you wanted to functionally disrupt a gene, you could cut it at that sequence, and then your DNA repair mechanisms are going to recognize that double-stranded break, and they're going to freak out, and they're going to fill it in with a bunch of nonsense. Therefore, what you've done is totally, stably disrupted that gene. Got it? You want to disrupt the gene? You devise this sequence, get in there, and cut it. And then every other cell that comes off of that will have that disrupted gene. Think cancer disruption. Another thing is, when you make this double-stranded break, it's got a little bit of a stickiness to its end. Let's say you wanted to insert a gene right there. So now I have opened up a site specifically. If I want to put something in there, I can do that. So now we have an edit function delete or edit function insert function that is very, very specific. Very, very cool. And that's another diagram about how zinc finger nucleases would work. So TALENs, transcription activation-like effector sequences, you can buy these too. So you can buy, this is like a huge Lego set. You know, you want to disrupt something, this is how it goes. And this was covered in a special issue of Science, or I mean of Nature recently, shown on this cover, sort of, this is like molecular scissors. And again, these are sequences that recognize, it's very much like zinc finger nucleases, and they recognize specific sequences, again, utilizing FOC1 as its nuclease, as its cutter, and is capable of doing this. I should mention that zinc finger nuclease technology is in whole owned by a company called Sangamo. You might want to look them up if they're not in your stock project, or if you have a little bit of coin you want to throw their way, that might be an interesting investment. I don't work for them. You know, I'm not proselytizing them or anything. So in recent reviews of TALEN technology, and this is like very recent reviews, here is how you could make knockout mutants in rats to study what would happen if you take out a specific gene function. We can do this in zebrafish. This is targeted disruption of zebrafish, showing you here in this figure which gene was targeted in this. What is shown in gray is what was filled in as the nonsense DNA to disrupt that gene function. And this has been done in human stem cells. Human pluripotent cells have been genetically modified for treatment, and we'll talk about stem cells during the medical biotechnology part of this. And we can insert genes like this green fluorescent protein, which glows under UV light and study its expression. And there's another diagram, a little bit more sophisticated, about what a talon-like nuclease system might look like. On both the 5' and the 3' end of that gene, you can see where FOC1 is going to cut and disrupt. And now, using that technology, you can buy knockout rats. I know you just need one. You need one of these for a pet, right? But if you wanted to order a rat with any gene that's been knocked out for studies that you just might want to conduct, you can purchase these. You can buy knockout mice. You can buy knockout zebrafish. Have at it. If you want to do it yourself, you can set up to make your own knockouts.